Hello, my name is Anirudh Viramala, and today I will be introducing the PIEZO1 protein. So, what exactly is the PIEZO1 protein? Well, PIEZO1 is a mechanosensitive ion channel that is found in the plasma membrane of cells. And in this video, I'll be telling you describing its structure and its function and how specific mutations affect the function of this protein. And I will try to show you as best as I can with the model I created. So let's get into it. So exactly, the piezo-1 protein responds to mechanical stress and pressure, and in turn converts it into chemical signals that the cell can respond to. So specific mutations in the piezo-1 protein, like the one we were assigned, was where the arginine is mutated into a histidine, which allows too many ions to enter the cell. And what does this cause basically? Well, this can cause a disease called hereditary xerocytosis, which affects red blood cell hydration and along with another disease known as generalized lymphatic dysplasia, which also disrupts fluid regulation in tissues. So let's talk about the structure of this protein exactly. So at first look, this protein looks kind of complicated, but honestly, each part has its own specific function. So these green blades that come out, these are the ones that actually detect the stress of the mechanical stress outside the cell that the cell is experiencing. And what happens is right now they're curved into this dome-like shape or a trimeric propeller shape. But as soon as pressure is exerted on the cell, these blades would actually open up and like expand outward, which is kind of hard to see on this model, but I can kind of like wiggle it out a little bit. So you can see it like moving out. And in turn, if you look inside, a canal kind of opens up, and then in a real cell, the ions would be able to pass freely through this molecule and enter the cell, making it lose like fluid or retain, and then causing to dehydrate a little bit. So exactly what I've used in this model is like I've used origami paper to simulate the blades, which kind of like anchor it into the surface of the cell. I use the cotton for the protein cap that kind of protects like these channels not to just for ions to just flow freely through. I used kind of like a toilet paper roll for the outside helix where the blades are directly bonded to. And then I use pipe cleaners on the inside, these yellow pipe cleaners on the inside for the inner helix. And then I use more cardboard for to use as beams which connect the blades, which anchor it to the outside helix. So in turn, if we try to simulate what it would be in a cell, the mechanical stress would cause the blades to open up, which in turn pull the inner helix open and then allow the ions to actually flow through the cell, flow through the protein and into the cell. So that's how exactly PIEZO1 works, and that's kind of how the mutation itself, like where arginine turns into histidine, that's how it kind of affects its function. So exactly, what is the exact mutation of arginine to histidine do exactly? Well, it kind of changes the actual properties of the amino acids itself, and then the properties of the protein. So changing arginine to histidine would actually keep the channel open longer than normal, causing the cell to lose more ions, which causes fluid to like kind of leave the cell and causing dehydration. So on a normal like on a cellular level, it can lead to a dehydration of like red blood cells. And then, but in an organismal level, it can lead to anemia and fatigue, which can affect overall health. So obviously with this model, it's not very accurate and to scale, but with what contemporary pictures look like on the internet, I use the materials that were available to me to try to like accurately depict what I can. It's kind of hard with the 2D model or trying to turn like a 2D image into like a 3D model, but this is what I've done so far. And I guess I have like these blades kind of sliding out, which when if in the cell when the mechanical stress is applied, which would open up, and I kind of have that demonstrated here where you can see the blades like slide out a little bit, and in turn would open up the canal on the inside. See, that's when the canal is open, and if let's say there was no like pressure or stress that was exerted on the cell, the blades would kind of close in like that. And instead, you can see how it's kind of like there's like it's more occluded on the inside, and there's more stuff in the way. And then we see like these anchors kind of get in the way on the other side. So, in summary, the piezo-1 is a mechanosensitive ion channel, and it uses mechanical stress to create cellular responses. When there is a mutation in this protein, it can cause serious diseases like anemia and affect like overall health. But with more research, I'm sure we can figure out ways to like combat the disease and combat the proteins and or sorry, combat the mutations and eventually improve overall health.